IB Bio Option B, Biotechnology, Bioinformatics Part 2, has its focus on the use of microorganisms in industry, penicillin production, citric acid production, and methane production. The essential idea is microorganisms can be used and modified to perform industrial processes. Here is the outline of all the movies available for Option B. Use this outline to find the movie you need for review. This movie is focused here. These 3 IB syllabus statements you've seen in Part 1, but they are an important foundation for Part 2. Microorganisms are metabolically diverse. List 4 metabolic processes carried out by named microorganisms. Microorganisms are used in industry because they are small and have a fast growth rate. Here is a curve that displays microorganism growth over time. You saw this curve in Part 1. I've included this curve again because it's important to remember that the rate of growth changes over time. Industrially, if you're interested in a continuous harvest of product, you would design the system to extract a continuous output of product while maintaining the population in the exponential growth phase. Some industrial cultures allow the microorganisms to reach the plateau phase or beyond. These are batch cultures where the product is harvested only at the end, then the tank is cleaned and cultures are started anew. Here are 2 new IB syllabus statements relevant to this movie. Pathway engineering optimizes genetic and regulatory processes within microorganisms. Pathway engineering is used industrially to produce metabolites of interest. Examples of products formed through microbial pathway engineering include alcohol, penicillin, citric acid, methane, otherwise known as biogas, and proteins of interest such as hepatitis B vaccine or antithrombin. Pathway engineering with microorganisms is the practice of optimizing genetic and regulatory processes within cells to increase the cell's production of a certain substance. These processes are chemical networks that use a series of biochemical reactions and enzymes that allow cells to convert raw materials into molecules of interest. Pathway engineering seeks a yield of useful products and seeks ways to increase the output in shorter periods of time for the purposes of increasing yield at a lower cost. Some of the common strategies used in pathway engineering are genetic engineering to increase the number of genes coding for the rate-limiting enzyme of the pathway. If enzyme 1 is rate-limiting, then to increase the number of enzyme 1 molecules would serve to increase production of the product. Genetic engineering that slows or blocks competing metabolic pathways. Genetic engineering to enhance the transcription of genes, specifically genes that might code for important enzymes, and to modify conditions for optimal enzyme action, pH specifically. Also remember that artificial selection is a mechanism to enhance metabolic pathways. Artificial selection is the foundational mechanism for much of modern biotechnology. In this diagram, a pathway is modeled, planned, and predicted. Gene deletions are made to reduce competing metabolic pathways. Gene insertions are used to increase the frequency of rate-limiting enzymes. DNA sequences are inserted to enhance the transcription of genes, and the output is monitored for successful production of product. In this slide, the product is 1,4-butanediol. It's used to make plastic fibers. 1 million tons per year are made worldwide. Each ton costs $2,000 US. Here are the next IB syllabus statements. Fermenters allow large-scale production of metabolites by microorganisms. Fermentation is carried out by batch or continuous culture. Define batch fermentation. Define continuous culture fermentation. I mentioned this earlier. Industrially, if you're interested in a continuous harvest of product, you would design the system to extract a continuous output of product while maintaining the population of microorganisms in the exponential growth phase. Some industrial cultures allow the microorganisms to reach the plateau phase or beyond. These are batch cultures where the product is harvested only at the end, then the tank is cleaned and the cultures are started anew. In continuous culture fermentation, the fresh nutrients flow into the fermenter continuously, and the product in the fermenter is withdrawn from the fermenter at the same flow rate of the input flow of nutrients. In continuous culture fermentation, the fresh nutrients flow into the fermenter continuously, and the product of the fermenter is withdrawn from the fermenter at the same flow rate as the input of nutrients. Continuous culture is superior to batch culture in several ways. There is no downtime to clean the tank. Microorganisms are small, they grow fast, and they can be maintained in the log phase of the growth curve. The success of production is easier to assess because there is a dynamic equilibrium of inputs and outputs. Batch culture fermentation is a technique where one or more nutrient substrates are fed into the bioreactor. The product is formed under certain conditions, temperature, pH, aeration, and remains in the bioreactor until the end of the run. The fermentation is stopped and the product is collected. Then the fermenter is cleaned, sterilized, to be ready for another batch. In batch fermentation, the fermentation is stopped at the end and the product is collected. Then the fermenter is cleaned, sterilized, as you can see in the left photo, to make it ready for another batch. This type of culture is typical of making alcohol using yeast or making penicillin. There are disadvantages for batch culture relative to continuous culture. There is downtime when the tank is being cleaned, and you can see this in the left photo. Batch culture is more complex than continuous culture because of the changing concentrations of products and reactants. pH is varying. It's a complicated mix of growing, dying, and dead cells. As well, with each culture starting fresh, it takes time before the organisms enter the period of high productivity. The growth curve on the right displays batch culture because it shows the product reaching a maximum as the substrates decline. Remember, fermenters allow large-scale production of metabolites by microorganisms. And can you define fermentation? Fermentation is the process in which a substance breaks down into a simpler substance, an enzyme-controlled chemical breakdown of an organic molecule, anaerobic mostly, but not always. The chemical breakdown of a substance by bacteria, yeasts, or other microorganisms, typically producing carbon dioxide and heat, but other products are possible. Here is an image of a deep tank fermenter. Inputs are microorganisms, nutrients, oxygen. Paddles mix the organisms and substrates and enzymes. Proper temperature is maintained. Conditions are monitored carefully by probes to ensure optimal conditions for the microbes and for the production of product. Here is a photograph of a deep tank fermenter. Conditions are monitored carefully by probes to ensure optimal conditions for the microbes and for the production of a product. Here is one last image of a deep tank fermenter. The substrates, the nutrients, are fed into the tank. pH is controlled. Temperature of the system is controlled. Airflow, oxygen, is controlled. Some fermentations are aerobic in the presence of oxygen, and some are anaerobic without oxygen. Here are 3 IB syllabus statements, the first 2 of which I've already spoken to. Conditions are maintained at optimal levels for the growth of microorganisms being cultured. Probes are used to monitor conditions within fermenters. And lastly, microorganisms in fermenters become limited by their own waste products. If wastes are not removed, then the rate of reaction is slowed. Wastes must be removed in order to maintain high rates of production. Here is a fermenter showing the inputs of carbon, the inputs of nitrogen and oxygen, temperature control, and the harvest of a product of interest. Wastes are being removed so that microorganisms are not limited by their own waste products. Let's take a look at deep tank batch fermentation in the mass production of penicillin. Penicillin production takes place in a deep tank fermenter using a batch culture process. In this photo, you are looking down into a clean tank. You can see the paddles that keep the mixture moving so that microorganisms and substrates are well mixed. Notice the ladder. The ladder allows someone into the fermenter to clean it after a batch of penicillin has been made. This is a limitation of batch culture. For a period of time, the fermenter is not producing product because it needs cleaning between batches. Penicillin is a secondary metabolite made by the mold Penicillium notatum. Penicillium notatum is seen here growing on the orange skin in the image on the left, and here in the micrograph on the right. Here is a deep tank penicillin fermenter. Notice the input of Penicillium notatum and sugar. Notice the oxygen supply. Penicillin fermentation is aerobic. Notice the control of temperature to maintain an optimal environment for the growth of penicillium, and the product is ultimately extracted, then the tank is cleaned to be ready for another batch. The steps in the process are quite simple. Penicillium notatum naturally produces the penicillin. Deep tank fermentation with sugars, it's a batch culture. Separate the penicillin, the antibiotic, from the culture and use the penicillin as an antibiotic in medicine. Here's a step-by-step process, probably a bit more detail than you really need to remember. A medium of Penicillium notatum, a nitrogen source, typically ammonia, and carbohydrates are added to the fermenter. After about 40 hours, penicillin begins to be secreted by the fungus Penicillium notatum. The mold cell matter is filtered from the product. Penicillin is extracted in an organic solvent in which it dissolves. Salts are added, and the penicillin precipitate is formed. This is washed and dried. Genetic engineering, otherwise known as recombinant DNA technology, serves to increase the number of genes coding for rate-limiting enzymes of a pathway, serves to slow or block competing metabolic pathways, or enhance the transcription of genes that would be important in coding for important enzymes. As well, it's important to modify the conditions for optimal enzyme action, and pH would be a good example. It's also important to remember the foundational technique in biotechnology, artificial selection. Choosing the most prolific individuals in a population to reproduce has been the most important biotech tool over hundreds, if not thousands, of years. Penicillin is a medical antibiotic industrially produced by continuous fermentation. One method of increasing the production of penicillin would be to genetically modify Penicillium notatum to express high levels of certain enzymes in the pathway, ACV synthase or isopenicillin N synthase or acetyltransferase. This would result in the production of higher concentrations of penicillin. For example, labs are working to modify a gene known as PENDE, which codes for acetyltransferase. Penicillin is a secondary metabolite of Penicillium notatum. It's produced when the growth of the fungus is inhibited by stress. It's not actually produced during normal growth. Production is also limited by feedback in the synthesis pathway of penicillin, and pathway engineering would serve to reduce the inhibition of the penicillin pathway. In the graph, you can see the increase in penicillin over time as the mold population grows and the nutrients are seen to decline. Penicillin amount increases dramatically at around 40 hours after the culture was started. Penicillin is a broad-spectrum antibiotic used to remedy bacterial infection. Now, let's look at the production of citric acid in a continuous fermentation culture using Aspergillus niger. Citric acid is important as a food preservative and a food flavoring molecule. You can see in the images on this slide, citric acid is naturally found in citrus fruits. Citric acid is industrially produced for various purposes, including to adjust the pH of wines. Here is the metabolic pathway within cells for the production of citric acid. The pathway should be familiar to you following the study of cell respiration and the study of the role of the mitochondrion and the Krebs cycle in cell respiration. Citric acid is an intermediate molecule in the Krebs cycle and can be extracted from cells in industrial processes. Succinate is industrially produced by continuous fermentation, and succinate is used as raw material in the production of citric acid. One method of increasing the production of succinate is to genetically modify E. coli to express high levels of formate dehydrogenase, FDH1. This results in the production of higher concentrations of NADH. The engineered pathway is shown here as a dotted line emphasized with a red arrow. A recent IB exam question asked specifically why high concentrations of NADH would favor the production of succinate, which ultimately would increase the production of citric acid. Increased NADH serves as a reducing agent in the production of succinate. Without NADH as a reducing agent, the pathway from organic intermediates to succinate would slow, if not stop. The engineered pathway, resulting in increased production of NADH, serves to produce more succinate, the desired product in the pathway. Citric acid is industrially produced and has various uses. Citric acid is used to enhance the flavor of foods, and citric acid is used as a preservative, as it would be in the making of pickles. Citric acid is used as a preservative of foods and for flavoring foods. 270,000 tons are produced per year worldwide. It's a $1.4 billion a year business. Here are micrographs of Aspergillus niger, the fungus that is used to industrially ferment organic molecules into citric acid. Citric acid is produced through continuous fermentation culture by Aspergillus niger. With continuous cultures, the level of nutrients and live cells are the inputs, and to maintain a continuous culture, the inputs enter the system at a constant rate, while the product, the output, must be removed at a similar rate to the inputs. You can see in this diagram that membranes are used to separate and recycle aspergillus while simultaneously removing the citric acid as it is formed. Continuous culture fermentation has the advantage over batch culture in that there will be little or no time lost due to startup and shutdown. The continuous removal of citric acid is critical. The accumulation of citric acid is inhibitory to the aspergillus. Continuous removal of product allows the fermentation to occur just below the citric acid level at which aspergillus is inhibited. Thus, the aspergillus cells are always viable and producing citric acid. Beetroot molasses and glucose are used as the feedstock, the substrates in the fermentation process. Nitrogen is added as ammonium nitrate. Iron accumulation would inhibit aspergillus, so iron is removed from the fermenter. Aspergillus hyphae are removed to prevent the mixture from becoming too viscous. And the pH is kept low, as you might expect in the industrial fermentation of an acidic product. These IB syllabus statements should feel comfortable. State that citric acid is produced by continuous fermentation by Aspergillus niger. Microorganisms in fermenters become limited by their own waste products. State that citric acid is used as a food preservative and for food flavoring. Provide examples. As we head toward the end of this movie, here is the next IB syllabus statement. Describe the production of methane, also known as biogas, by prokaryotic archaeans from organic matter in fermenters. Here are micrographs of archaeans that produce methane gas, CH4. Archaeans that produce methane are known as methanogens, and methane is also known as biogas. All of life is divided into three domains: eubacteria, archaea, and eukarya. The archaeans are prokaryotic cells with certain characteristics that are similar to eukaryotes. You can see the methanogens here, here, and here within the archaean domain. Methanogenesis is an anaerobic fermentation. Oxygen inhibits the growth of methanogens. Carbon serves as the final electron acceptor in the last step of organic matter decomposition. Methane is produced as intermediates are decomposed. Here are anaerobic methane production pathways in bacteria, starting from organic solids such as coal through to acetic acid to methane, outlined in this pathway, or more directly from carbon dioxide to methane, outlined in this pathway. Increasing the quantity of rate-limiting enzymes or enhancing the transcription of genes that code for enzymes or coenzymes would serve to produce greater quantities of methane. Slowing the rate of competing pathways would also serve to increase methane production. Sewage and organic wastes such as manure, compost, landfill material can be the substrates that are fermented through acetic acid as an intermediate to produce methane. The methane can be withdrawn and used as an energy source. Organic matter is fed into a fermentation tank. Under optimal temperature and pH conditions, the organic matter is decomposed into biogas. Wastes are removed to maintain optimal conditions. Again, organic material, animal waste, food waste, agricultural waste, manure, grasses, are fed into a digester. Methanogenic bacteria decompose the organic material in an anaerobic environment. Methane, biogas, is extracted. Biogas can be used to generate electricity or heat. Biogas is an energy source, and when derived from the decomposition of organic matter, like manure or grasses, it is a renewable energy source. Methanogens live in the guts of mammals. Methanogens anaerobically transform organic molecules into methane. If the feedstock for methane production is organic, as it is here, then the production of methane is renewable. Here is a diagram of a small-scale methane production system that emphasizes the renewable nature of methane production from organic matter. Carbon dioxide is taken up by plants from the air through photosynthesis. Plants are taken up by the animal. Manure enters the fermentation tank as feedstock for anaerobic methanogens. The biogas is collected and used to generate electricity or heat. The carbon dioxide released from the combustion of methane returns to the atmosphere, where it is taken up by plants once again. Here are photographs from a U.S. farm that collects methane from its cattle and heats the farm buildings by burning the methane. So at this point, could you describe the design of a small-scale fermenter in the production of biogas? Here is a photograph of a small-scale fermenter producing methane gas, biogas, in India. Here is a photograph of an even smaller system producing biogas that fills the tire. Start with organic matter, ensure a dose of methanogenic archaeans, provide the proper temperature and pH, and you could fill all the tires you have. Or maybe you could heat the building. Methane is a fuel, an energy source. Methane, produced using organic matter as the principal feedstock, is a renewable energy source. With the threat of climate change, industry is looking toward numerous species to serve in bioreactors, producing organic matter that could be used in fermentation tanks to produce energy, renewable energy. In this slide, we can see two species that are being genetically engineered for the production of biofuels. Alcohols or other organic intermediates, such as pyruvate, are produced from organic matter, and they can be used for energy. To meet the energy demands of future generations and to reduce the threat of climate change, industry is looking to the microbial conversion of renewable feedstocks for the production of fuels for energy. Saccharomyces cerevisiae, yeast, is a robust organism with a wide range of pH tolerance and a simple nutrient requirement. Genetic engineering, along with artificial selection, has been successfully used with yeast and other microorganisms to extract pyruvate or malate from yeast fermenters rather than alcohol. These organic intermediates are more versatile energy sources in the bioindustrial production of energy. One question remains: will we use our farming system to grow food or renewable energy for our cars? 8 bushels of corn is 22 gallons of ethanol, or enough food to feed a person for a whole year. And that brings us to the end of Option B, Biotechnology, Bioinformatics, Part 2. We have had a focus on microorganisms and industrial fermentation in this movie.